Longitudinal growth during the fetal and neonatal periods involves depletion of chondroprogenitors, whereas later in life, chondroprogenitors acquire the ability to self-renew, resulting in the formation of large columns of chondrocytes. At the same time, these chondroprogenitors begin to express stem cell markers and undergo symmetric cell division. Hedgehog and mTORC1 signaling pathways regulate these self-renewing chondroprogenitors. Findings of the study show that a niche for stem cells develops after birth in the epiphyseal growth plate. The growth plate provides a continuous supply of chondrocytes. Key terms: chondroprogenitors, cells that will become chondrocytes. Chondrocytes, cells that are found in cartilage and produce and maintain the cartilaginous matrix, which contain collagen and proteoglycans. Hedgehog signaling pathway promotes proliferation of adult stem cells. mTORC1, mammalian target of rapamycin complex 1, modulates balance between symmetric and asymmetric stem cell division. Epiphyseal growth plate, site of longitudinal growth of long bones, responsible for new bone growth, located at each end of a long bone.
To characterize the generation of epiphyseal chondrocytes, scientists use clonal lineage tracing of crossing CoL2 Cre-ERT mice with multicolor R26R confetti reporter mice. Recombination was induced by a tamoxifen injection on postnatal day 3. 2 days after the injection, all types of the chondrocytes were labeled as seen in Figure 1A, whereas 1 month later, only stable clones originating from long-term progenitors in the resting zone were observed, as seen in Figure 1B. To assess growth plate dynamics, tracing of chondrocytes for 10 or 14 days at different ages revealed that only clones with few cells during the fetal neonatal period increased markedly later. Clones were initially small in bones labeled at both P3 and P30 and expanded with time only in P30 bones. Growth plate columns were multiclonal at the fetal neonatal stage but became monoclonal after P30. These monoclonal columns remained in renewing cartilage for at least 5 months. Multiclonal neonatal columns appear to arise from several progenitors, whereas monoclonal columns originate from a single stable progenitor. Scientists observed that the neonatal patchy pattern of clonality changed to stable monoclonal columns in mature mice, suggesting the presence of a subset of progenitors that generate large stable clones in mature mice. Scientists also developed a model to understand the shift in clonality. This model indicated that obtaining long, stable monoclonal columns requires the uppermost cells to undergo renewal, whereas stacking of short clones requires constant recruitment of round cells into the proliferative pool. These observations led to a model in which the pool of chondroprogenitors is slowly depleting by direct recruitment during fetal neonatal growth, whereas after the growth plate forms, remaining chondroprogenitors acquire the capacity of self-renewal, each producing many progeny.
The idea that chondroprogenitors are consumed during the fetal neonatal period and gain the ability to renew later in life is supported by our research on the secondary ossification center, aka SOC. Now, the SOC itself eventually matures into the epiphyseal bone layer, in which the very top clonal cell in the plate itself is in contact with the bone. Now, combining laser dissection and single-cell RNA sequencing protocols, we were able to determine that as the SOC matures, ECM genes are upregulated and these cells are organized in a manner which ultimately results in a niche being formed. Now, during this process, progenitors expressing the double positive for CD73 and CD49E expand, form colonies, and ultimately can differentiate into things such as chondrocytes, osteoblasts, or even adipocytes. Once a niche is established, the clones gain the ability to renew using a combination of both symmetric and asymmetric division. Figure 2 provides visual evidence regarding the claims of niche establishment resulting in chondroprogenitor renewal. Sections A through C depict the positive effects of Salk development on the clones themselves, and as shown by the graph to the right, clone size increases with proximity to the Salk. Section D, however, shows what happens when the mice are administered exinitab, where Salk maturation is delayed, resulting in reduced clone size. The remaining sections confirm the presence of CD73 and the directionality of clone division itself.
So as we figure out what factors decide the stem cell renewal, let's look at how the cell division are determined. We learned in previous lectures that the cell have both symmetric and asymmetric divisions. Symmetric divisions help to maintain the cell's own growth, but asymmetric divisions help the cell to differentiate into many cell types. What actually determines whether your stem cell will undergo asymmetrically or symmetrically? In this article, I hypothesize that activation of mTORC1 gene signaling can change the cell from asymmetric to symmetric divisions. In this experiment, the designer activated the mTORC1 by chondrocyte-specific ablation of TS1 gene, C1 gene. However, there wasn't any expression of chondrocyte markers, indicating that asymmetric division is not possible here. But there was an expression of cell marker CD73 and a PAR3, and also the number and thickness of multi-columnar clone in these mice increased with time. All of this together can prove that mTORC1 does decide how the cell divide.
A stem cell niche develops in the postnatal epiphyseal growth plate, which allows for the self-renew of chondroprogenitors and a continuous supply of chondrocytes for bone growth. The significance to regenerative medicine is that this study may provide new information to help improve the treatment of children with bone growth disorders.